I'm gonna be looking at the sponges and corals more than I am the fish.
I'm happy to hear it.
I was going to say, you probably just made Megan very happy.
Yeah.
Yeah, because, you know, when we're spearfishing, we don't really think about it. But
Yeah,
just
you're
being here,
focused.
yeah, being here, getting to know all these different species and animals, and it's got me to appreciate it a whole lot more. The anemones.
Yeah, sometimes when I'm in the water snorkeling or diving, I pay so much attention to all the corals, and I don't pay as much attention to the fish. I have to remember to look around and be like, oh, wow,
There's fish.
fish everywhere. That was the most amazing part of the dive I did the other week.
Do we have two fish scientists downstairs cringing at this?
Well, I'm like focused in, well, Jeff was my dive buddy, and I was like focused in on looking at some of the corals, and then you
He's
point
focused on
out
the
some,
fish.
yeah, like these like huge fish, big schools of barracuda, all this like cool stuff, and I'm like, oh yeah, wow, this is amazing. Have never seen anything like it.
Yeah, perfect dive buddies.
Yeah, compliment each
Yeah, I was
other.
going to say it's a good balance.
Was this, you guys were diving in Palau?
Yeah.
Do you remember the name of the dive?
We went to German Channel, Blue Corner, and New Drop Off, and the big schools of fish were at the New Drop Off site.
Yeah, the drop-off reefs are really pretty and also good for what you said, where on one side you'll have like a wall of really amazing like stuff and corals and sponges, and then on the other side you have the pelagic ocean, so you can kind of look out for sharks or pelagic fish or schools of fish even. So
yeah,
those are some of my favorite dives too.
It was just so amazing. Like there was this huge Napoleon wrasse that kept coming around, and like big schools of barracuda, and just like so many fish. It was unreal.
I'm glad you guys enjoyed.
I'm looking forward to some more Palau exploration time.
Two 0-155. Oriel's voice sounds different.
You mean Flavio's voice?
Flavio's voice, yeah.
Yeah. I guess Chris is done with his post nap situation.
Now what's that fish?
Cuskeel?
Yeah.
Nice.
Oh, a bigger one.
Yeah, that one is a bigger one.
My goal is to make Megan proud.
I'm a little better at the coral than the fish.
That's fair.
Yeah.
Me too.
The fish all look a little samey to me. So what is the difference between a fish and an eel?
Well, so like we call this a eel because of like the elongated body.
Right.
This is a better question for Jeff. their body form, the eel's body form is a little more skinny and long, and tapered. There's probably a lot more to be said about it that I can't articulate properly, seeing as I'm not a fish biologist.
Yeah,
So is it
for sure.
an eel is not in considered
So
a fish?
all eels are fish.
Okay, but not all fish,
Fish
are
are
eels.
eels.
Yeah.
Okay.
This is a really nice
Little
area.
outcropping.
A lot of color.
Right at waypoint 4.
Let's hope the other waypoints deliver.
Yeah, exactly.
Oh, this like ridge feature at the top looks really exciting. I'm super stoked about
And
it.
we're hitting that on our watch?
No, I don't think we're gonna make it there.
No.
Yeah.
No, but we'll be back.
But we'll be back.
That's
We'll
true.
be at the ridge at the end or at the beginning of our watch, and we will have decided either we're going to transverse this ridge feature, or we might jump off the side and go up the wall. But it's super skinny. It's like, I think only 200 meters across. Or
It will be the
maybe
last
it's-
watch of the expedition.
Yay.
yeah, it's like
Is
really
that an
small
urchin?
feature.
Yeah, I think it's
Yeah,
the
there's
urchin on
another
the
one of
right.
those sparrow-somatid pancake urchins. I've been seeing quite a few of those.
This is
Yeah,
really
so-
nice.
Yeah, so much to look at here.
Can we zoom in on the yellow? It looks like it might be zoanthids.
Yep, push in there.
Yeah, almost
Looks
from afar
odd.
looked like gold coral, but
Yeah, well, I mean, gold coral is zoanthid.
Yeah.
So, yeah. Yeah, but this is way out of its depth range to be
yeah.
gold coral.
Got some snails.
Oh yeah, there's
So-
a lot of associates. There's some gooseneck barnacles, hydroids, squat lobsters, anemone, foraminiferans,
Okay,
brittle
come on.
stars. And then there's a pseudomunida on the back side.
Oh, I love the purple. The purple corals are just stands out to
Yeah,
me.
they're great color. All right, we've made our way to waypoint 4, so we're gonna hopefully head up to this pink peak of waypoint 5, sort of a local high.
Nice. So between waypoint 4 and waypoint 5, we'll pass by our 1,000 meter mark. So we'll do a Niskin there, I guess, right, Elsa?
Yes, please. And then I will be there this morning to collect it. Blodak Blodak has really been assisting a lot, even though he's not technically here as a on the science team. He has been there for almost every eDNA collection. So shout out to Blodak in addition to
Shout out.
sitting watch, doing interactions at all times of the day, and assisting with the eDNA.
Yeah, no, I love it. It's a good experience to kind of just understand the different roles of the ship and how it all comes together.
Now I know why people refuse to stop calling you the principal.
Yeah, those kids.
Multitasking expert.
Yeah, so in the chat, Kenneth Sulak is just talking also about the something that differentiates eels is that they have a larval stage in the midwater, so the upper 200 meters of the ocean. And he says their larvae mostly do not feed and live several weeks to several years before beginning to transform to the adult stage.
Mm.
Oh, interesting.
Yeah.
Thanks for that fact, Ken.
He called them epipelagic dialysis bags. So absorbing dissolved nutrients. Oh, interesting.
Is that an anemone? Or an urchin?
Yeah, I think an anemone, but we'll see.
It's an anemone.
Yep. Let's do a quick snap zoom.
Push it there. And this is a bamboo that's
I think it's a black coral in front of it.
it's a long and skinny one.
Is it
Yeah.
Stichopathes?
Yeah.
Looks like. We collected one of those the
Yep.
other day.
That's great. Thanks, Jake.
Think about.
Yeah, the scientific names of all groups of black corals will have pathes in there. So different genera, there's about 300 or so described species of black corals or antipatherians. The name there, antipathes, comes from the Greek, antipathogen, or against evil, against disease, as in old Greek and Roman culture, but also in Chinese culture and in many Pacific Island culture, a lot of remedies made out of black corals, and that's the root of that name. And so that's stuck, and so all the genera and families that were subsequently created all have that pathes still in the name. And yeah, this common, like the Gorgonians too, will have gorgia as part of their name. So in a lot of groups you have those roots directing back to something about this describing that species or that group of species.
I'm loving the taxonomic etymology.
Yeah, I find it's easier to remember that.
If you understand what it means.
Yeah, it has some story behind it. And
definitely.
but it's also something that just for me as a transplant, and I moved to the Pacific Island region in my early adulthood, and a lot of Pacific Island culture, genealogy being so important, and origin stories, and that's being so important and so culturally significant, and that the process is so important. It's in many cases even more important than the outcome. And yeah, it's something I've gained deep appreciation, and then it's always wonderful when you work in different places across the Pacific Island region and seeing how many of these cultures have some similarities in terms of these values, which I find incredibly incredibly fascinating. Yeah, we got a rat tail fish off screen.
Might be too big to collect.
I think so, yeah.
Yeah.
For sure.
Sorry, Jeff.
Yeah, it's really interesting that you bring that up, that the process is almost more important than the outcome, because that definitely resonates. I think with thinking about working within Pacific Island cultures and the Palauan culture, have relationships are so important, and almost who the person is and their relationship to you is just as much as important as what they know or what they bring to the table. So, and for sure OET has been really great about building those relationships for the past 2 years, not just sort of coming in with parachute science, so to say, to do the work and then leave and not have any lasting sort of impact for the community. And I think that's the opposite of what will happen here. And yeah, thanks for that, Dan.
Well, thank you for those kind words. Yeah, it's truly what we hope, and it's, you know, this is hopefully the start of many great things to come. And yeah, the relationships are so important. And oh, there's a smaller rat tail.
It looks like the...
Collectible?
Yeah, I think it's really interesting how the way that we see each other and the way that we see what we do often kind of mimic each other. So when we start to appreciate relationships as like the most important part of the work, we also see a way of speaking about the environment through relations and interconnections, and instead of a kind of dropping in or parachuting or objectifying of the natural world or each other. So it's really, yeah, it's really inspiring to see the kind of evolutions of practices and collaborations take place, and the models be created and co-created. It's just feeling very grateful to be a guest in these waters and to, yeah, to learn so much from the deep sea inhabitants, and to learn so much from all of each other and our partners.
Anthomastus?
Yes,
Yeah.
I think so.
Yep, that's the Anthomastus tachynotus.
Tachynotus.
Yep, it's got the long body.
Oh, like the part that attaches to the
Yeah, like
ventos?
that base part is really long and skinny.
So other species would be
Is
like
that
sort
better?
of more squat.
Okay.
Yeah, and tend to be more pink, and the tachynotus tend to be a little more orange.
Oh.
That's not the real character, but like, just seems to be on average how they look.
Right. Experience is the best teacher. And I know Megan spends a lot of time
Annotating.
annotating.
Yeah, lots and lots of time looking at these animals, and when we get back to shore, we'll be annotating this these dives starting next year. We're currently finishing up some work that was done last year in the Papahanaumokuakea Marine National Monument, the NA154 cruise.
154.
Which Elsa was
Oh, that's the
was
one I
on.
was on.
Yeah.
And you remember how many corals there were, don't you?
Yes, yeah,
It was,
yeah.
it's a doozy
Yeah.
annotating that.
Yeah.
There are some dives on these 2 Palau cruises that are going to be just as challenging. But it's going to be really nice to be able to annotate this work and make it available to everyone who's interested in learning more about the ecology of the deep sea environments around Palau. And we can also use the data to compare to other places that we've been, see how they're similar and how they're different, look into bigger biogeographic patterns, and learn more just in general about our deep sea, as well as specific things like new species and new occurrences of animals in this region.
So will your annotations be available online?
They
Are
will.
they published on the DARC website?
They will be published on OBIS
Okay.
and on the NOAA Deep Sea Corals portal, and so they will be available to anyone around the world.
Perfect. Yeah, well,
So it's going to take us a while to actually get everything annotated and fully completed, but that will be our goal for next year.
yeah, thanks, Megan. So I'm certain we will
Gonna move the
probably
ship.
Yep.
make use of the annotations and just to get really like a quantification of the species across a lot of the seamounts that we've been seeing. And then along with the annotations, do you do density calculations at all, or just the annotation? Mm-hmm.
Megan, I don't think you're on SPO.
Oh, okay. Yeah, I'm just talking to myself right now.
Well, I could hear you.
Yeah, sorry.
It was beautiful.
You said a lot of really nice things.
We all could hear you.
Yeah, yeah. Anyway, so yeah, density calculations. You can definitely use our annotations to calculate density of animals. So the annotations include identification and counts of every single organism observed, and you can estimate the area surveyed based on the camera. We have the lasers there that are 10 centimeters, and you can calculate the area that we're viewing and multiply it by the distance covered to get a really rough estimate of the area that we've surveyed throughout our dive transects. And you can then estimate a density of organisms and abundance of organisms across this dive transect. That's what I've done in the past for the work that I did for my thesis research.
Cool, yeah.
Yeah, and this is an Enelopsamia.
The little unbranched one, or the big one? Yeah, okay.
Yeah, the little one is a little primnoid coral. It's unbranched primnoid. And there's a little squat lobster on it.
Yeah, that's great. Thanks, Jake. And
Okay.
if we can also just quick snap zoom on the sea star to our left. I believe that's Henricia. Megan, is that right? Or
Could be Henricia, yeah. I can't see it well enough, but that is a great guess. Yeah, it looks like it. I think you're right.
Push it there. Oh. Sorry.
wonderful, yeah. Yeah, so the work that Megan and others in that lab will do will, there's a lot of different things that you can get out of that. So obviously first just a list of species, just increasing the inventory of the close to 3,000 marine species we know in Palau. There will be many more added to that just by exporting all the species there. Then the densities, you can get something, but then you also can start looking at the spatial distribution of different species and patterns and vertical and horizontal patterns. So both vertical. We'll have a complete cross-section here of about 500 meters to about 32 meters, 3,200 meters. Well, we've done with this now 13 dives going across a wide depth range, and it's trying to understand a little bit of the spatial distribution of what are the depth ranges of the individual species, but then also what are the geographic distributions. I think just from a, this will require some statistics and some analysis, but just from a, having now seen these dives, the southern and southwestern seamounts do look a bit different than what we saw around the north. And yeah, all very interesting, and just showcases the importance of these sanctuaries and large-scale marine protected areas. There's natural laboratories that allow for so many different scientific questions to be answered. And the really key factor there is because they are protected, you have isolated many of the human impacts that you do have close to cities or close to human population center. Those have been removed. They have not been removed completely. Obviously, climate change and other things are still impacting things at a large scales, but you've removed some of them, and this gives you a great window into, into the past, if you will.
Yeah, this is great work, and it's really appreciated, especially to understand the National Marine Sanctuary and everything that's within it. And I think it's also interesting how during one of the presentations they showed the food web and how it's all connected. So, and I think that was from your research.
That was Jeff.
Yeah, Jeff's work. Yeah.
Yeah, the really interesting fact that stood out from his presentation is that when we're eating pelagic fish, we're only one step removed from sort of these smaller organisms, the microorganisms living in the midwater and in the deep ocean. So that's a really striking way of framing how closely we are connected to this natural environment that we don't necessarily see all the time or think of.
Yeah, absolutely, and that's again one of the things that I've really enjoyed about having moved later in my life to the Pacific Island region, where that's common sense. People just, the ocean is part of everyday life. Like, it's something that is just, the ebbs and flows and all these things, these natural rhythms of the ocean is something that it's well within the culture, and in many other parts of the world where you're isolated from the ocean, you don't realize those things. And
Yeah, I don't think I could ever live in a place that was isolated from the ocean. And I never have, and I don't plan to.
yeah, it's funny, I grew up in the mountains. I was born in a city that's 10,000 feet high, and I had to drive many hours to get to the ocean. And yeah, it does have very powerful healing power, and it's captivating, and once you see it, it's just and experience it, it's just a way of life.
Yeah, I totally agree with that. I grew up in the Midwest, so not a lot of ocean there. Everybody's like, oh, the lakes, but it's not the same.
Yeah, isn't it? I mean, I'm probably wildly wrong with this figure, but isn't it like 70% of human settlement is across like coasts and near the ocean? Probably wrong, but a large figure like that.
Yeah.
A little bit of a current. I'm like all the way lateral to port and just creeping.
Yeah, left to right.
Yeah.
Coming from 045.
Yeah, I think so. Northwesterly, coming around northwest.
Yeah.
Yeah, that's a good thing.
It's picked up a bit in the last 100 or so meters of water depth.
So that would basically put this current right parallel to this ridge feature that we're basically.
Yeah, that's what I was sort of expecting to see here too, because this ridge is almost running north-south.
Yeah, I think having lived near the ocean for quite a long time and spending the last decade going to sea, it was a change to move somewhere not directly on the coast, but I can see a canal from where I live, and I spend my time often by other bodies of water, and I think it's brought a bit more appreciation for all the ways that water bodies are interconnected, be it rivers or clouds or oceans. And I think it's also helped me appreciate new ways to speak about how the ocean actually connects all of us, even if we're not directly living next to it.
Yep.
I never thought of clouds as a water body, but I guess they are.
Yeah, I mean, I guess it's a bit open to your interpretation, but I think, yeah, there's different ways to, I guess, scientifically refer to water bodies and poetically refer to water bodies, humans as bodies of water as well.
Oh yeah, definitely.
But I feel like clouds are one that most places in the world, everyone has access to sky views, albeit different different kinds of skies, and there's a lot of water up there.
Never thought about it that way. Thank you for
Reframing.
Mhm.
We got a lot of really nice clouds out here too.
Oh yeah, we've
In
been
the-
getting really cool,
yeah.
really huge cumulus and some really high up cirrus clouds as well, making beautiful sunsets.
Let's do a quick snap zoom here on that glass sponge.
Zoom in on it.
This one looks a little bit different than,
It's a Faria. Yeah,
Still a Faria.
Yep.
kind of looks different than the other ones.
Yeah, well, it's not. It's like the Faria Oka, not like the other Farias
That's
that
great. Thanks,
that
Sarah.
were more scoop
Yep, come
shaped
up.
that we're
Thank you.
seeing all over the place. I think this is a different species.
Cool.
I was also gonna guess Fariidae, so I'm happy that it was
Yeah,
correct.
you're right.
Megan, can we get to just estimate range to waypoint 5, please?
Of course, yes. We got 380 meters.
Roger that. Thank you much. So moving right along here, should be, we're about a kilometer and a half now from where we started this dive. But still about 2 kilometers where we hope to end it. We still have a good amount of dive time left on today's dive. So we'll be back on deck at 2 a.m. Palau time.
So, siphonophore. One view.
Yeah, coming right up there.
Oh,
See if
yeah.
I can catch it.
Oh, I don't think we're gonna catch it.
No.
It's already breaking apart.
Oh.
Ah,
Ooh.
Oh, here's the,
sponge.
yeah, there's the,
it's the Caliphacus.
Caliphacus.
I'll zoom in. Do a quick little snap zoom of the sky.
So these can have multiple sort of bulbs or just
Oh, yeah.
single?
So there is a species that has the multi-puff, lots of puffs. And we saw one on the 167 cruise that had 10 puffs.
Oh, wow.
Yeah, I'd never seen, normally it's like 3 or 4.
All right.
So 10 was a little excessive. I had never seen that before. It was very impressive.
I think I was watching that live while you were watching that sponge with 10. And you were kind of, yeah.
Yeah, that was a very neat one. Definitely a new observation.
So yeah, we should have just a little under 11 hours of bottom time remaining on today's dive. The plan is to end at the summit of the seamount, which has a depth of about 700 meters, so it should be about a half an hour ascent towards the end. We'll secure the ROVs on deck then at 2 o'clock and then start our 26-hour transit back to Malakal Harbor, conducting mapping operations along the way, as well as slowing down about halfway in that transit to collect our final surface eDNA sample set. And that will then conclude the second of Lebu's voyage expeditions to explore the Palau National Marine Sanctuary. EV Nautilus will then be docked at Malakal Harbor for the next 3 days before starting the... Yeah, let's take a zoom.
Is that predation?
Yeah.
Looks like it's getting quite a lot.
So this is a type of cookie star called Hippasteria, and it's feeding on this Calyptrophora wyvili, the primnoid coral.
Zoom in. Just try and hold it here a little bit.
So these sea stars are known to be coral predators. They feed on corals, and this is a nice smorgasbord here for it. They will feed by extruding their stomachs out of their bodies and around what they're trying to eat. So as we zoom in, you're going to be able to see that. It's real weird.
Oh, moving back a little. Sorry, guys.
Current strong.
Oh. Yeah. All right. Yeah, I got like the little corner of the porch on there and was trying to keep her steady.
Can we take just a zoom on this?
Yeah.
Please?
Is that coral rubble?
Yeah, I don't know. It looks like a dead something, but very... Jeff and Greta mentioned seeing a few bizarre looking dead things that were large. I don't know if this is what they were referring to.
This is a dead Tritoplura.
Okay, can zoom in there.
Tetrito plural.
Tiny cup coral too on the bottom. All right, thanks. Thanks, Sarah.
All right, come out.
Yeah, maybe let's do a quick on these double sea star plus coral combo.
All right, zoom in.
We got an urchin.
It's a really
Yeah,
big.
so we have a pencil urchin and Enelopsamia coral.
Mm.
That looks dead. And then Henricia sea star. And
What looks dead? I'm sorry.
The coral.
Oh,
Yeah, that doesn't look alive anymore. The Henricia sea star and the Benthopectinidae sea star. So the Benthopectinidae is the pink one, and the Henricia is the white one.
Reminds me of when I'm sunbathing with my daughter at the beach, just laying down there next to a palm tree.
Okay, come out.
Is this dead?
Yeah.
Yep, dead bamboo coral.
Oh, I
Did it
think
cry
only
now?
the tip is dead. It still has polyps.
On
A
the
little
lower
bit.
part.
Zoom in.
Okay, I kind of see them now.
Are they polyps or?
Yeah, no, it's alive.
It's just
Yeah.
the tip that's kind of dead.
Yeah, the tip,
And
the tissue is receded from it.
And the stock. Huh.
So we are approaching our 1,000-meter mark where we'll try to get our Niskin sample. It's about 30 meters to go. A few more of these pancake urchins. Acantogorgia.
That's a nice bolosoma.
It is.
Which basically, back to
Okay,
the
zoom in.
names, Bolo and Soma, round body,
Mm.
origin of those words.
It is round.
can come out?
It's kind of tuck in the arm.
Thank you.
I really enjoy all the different shapes we're seeing from the sponges.
Oh yeah, they're so much fun. I say this quite a bit, but I feel like a lot of these deep sea sponges would make really great like IKEA style housewares.
I was gonna say, inspired.
Couches. Sponge couches.
Yeah, sponge lamps, you know.
Lamps.
You could sponge everything.
I'm sure we could design a collection.
Make like stools.
I like those, the nematodes would make great little stools or poofs, ottomans. The Lophocalyx would make like a great comfy couch.
Yeah.
Thank you.
When we're looking at the sponges in the lab, both Noel, our OET science coordinator, and myself are developing quite a passion for sponges. I think we are sponge enthusiasts, and a lot of encouragement to any other sponge enthusiasts to follow your passion, since there's not enough sponge experts out there.
And so much to do yet.
Yeah.
Way over 10,000 species,
Wow.
and very few of them actually well understood.
Just in the deep sea.
Yeah, that's just total. Yeah, and sponges are found in all waters, not just the ocean, and a quite diverse group. Let's see. What do we got there?
Fish.
Yeah.
Alright, we're starting to see a little bit more fish.
Zoom in.
Okay.
Let's see if I can.
Yeah, that current's really
Somewhat.
going, isn't it?
It is. I've had to, if I go into it like diagonally forward to port, I can get it. But if I go straight port, we don't really move.
Yeah.
Let me back up a little.
Looks like Bathygatas.
Which is a rat tail.
I don't know.
No?
Well, I can't see the filaments. Oh, look at the mouth. Oh.
Is that open?
Yeah, it opened its mouth.
Oh, there it is again. What? Oh, okay. So it kind of extends out.
Yeah.
Is it feeding?
I don't know.
Oh.
It's yawning. It's bored.
Yeah. There it goes again.
Interesting behavior.
Oh.
Yeah, not Bathygatas.
Jeff,
The
Jeff is
chat.
interested.
Yeah, Jeff. Well, maybe this will work if you have a different, if you have, Sarah, you fly and Jake, you like
Yeah,
arm it in.
I'm willing to
Okay,
try.
I'm gonna zoom out.
Yes, please. Awesome, thank you. Man, he is fast.
Yeah.
Cruising down here. Wait.
Jeff thinks it's maybe a Nizumia.
It could be. Yeah.
Mizumia.
It's right up here. See if I can cut him off.
Nice. Oh.
Mute my...
Oh no!
Oh no!
Oh, it was
Alright,
almost
let me
there
try.
too.
We're on the edge of our seats back here with our mics muted
I was gonna say, I was
to be
holding my
not
breath.
distracting.
That's all right. I still see him. I'm gonna try and catch up here. Just fighting the current a little bit. Let's turn this up.
You got this, team.
How far can that thing extend? As long as the arm?
No.
Oh.
All right, almost there.
Okay.
Tricky. Oh, very tricky. So fast. Oh no. Go forward. Forward. I am like full
Thrust.
thrust right now.
Yeah.
All right, I think he's coming back.
Yeah.
Yeah, I don't think it's gonna happen.
Yeah, I think so.
Not unless it was like perfectly.
Yeah, like the head is perfectly aligned and it
Yeah.
just couldn't.
Jeff says thanks.
Yeah.
It was a much bigger fish.
Yeah,
That was a good effort.
It's good teamwork.
it's very tricky.
It's fun to try.
Yep. Thank you for the assistance.
Jeff says that he really appreciates all the effort, so.
Yeah, we're here holding our breath. I'm wondering how he's feeling.
Yeah.
I didn't quite realize how large it was until it came really close to the slurp, and I was like, oh.
We got some viewers that were also out of their seats.
How many viewers do we have with us at the moment?
Check real quick. So we got about 44. Yeah.
Nice.
You got people from Germany, Guam, Japan, Netherlands, New Zealand, Sweden, Australia, United Kingdom, Canada, what's up, Palau, and then the US. We are currently 1,018 meters down here, working our way up the seamount.
This is quite a current we're going into.
Yeah.
Yeah, getting a lot of current over here.
Yeah, I know at least one of the Palau viewers is our head researcher, Igalau. She sent me a screenshot she took of the pelagic cucumber we saw earlier.
Awesome.
So shout out to Igalau.
Definitely. I'm sure. I hope our pelagic sea cucumber makes the highlight reel.
Yeah, the Chanacops I saw.
Yeah, the Chaunacops made it.
The Chanacops always
Can,
makes
can we
it.
just do a snap zoom?
On there? Yeah. All right, go in.
Looks like a Madrepora.
Yeah. Which is different. But
Oh, little fish.
That's a little cyclothony. I got our favorite carrier crab. This one looks like it's carrying some zoanthids.
Zoanthids.
Yeah.
That's a first for maybe this expedition.
Yeah. First zoanthid carrier.
Yeah, you can tell the current is, but I'm guessing that these organisms kind of enjoy the current.
Yep,
More
more
than we
current
are.
means more food.
Yeah.
yeah, I'm just trying to, if we're in a good enough position, like I know this is going to be extremely fragile, but if there is a opportunity to collect this, I don't know.
How you feel, Jake?
Yeah, it's,
Yeah, let's try it.
I mean, it would be basically trying to get this
All right,
with
I'm
the
gonna come
base,
wide.
but it's
Can
going to
come
be
wide?
very, very fragile.
Yeah.
Wait till you can get
See if I can
steady
yeah,
there.
I get nested here if I can.
If it goes well, we have our forward lambda box available. That would probably be the easiest.
Okay, I think. I'm good.
So going for the
The
White-
white
white?
one,
Yeah.
yeah.
On the bottom, or
Yeah, but I have a feeling they're actually all somewhat attached, but yeah, the white kind of hard, and it's going to be very brittle. So if you can grab one and maybe the very gently, the thickest piece.
Want
That's
to push in
gonna
a bit there? Tilt down a bit.
All right.
tilt down a bit.
How's that?
It's good.
Okay.
Is that the base behind it? Right?
Yeah, the base
Oh.
is behind it. It's kind of, it had fallen over at some point and it started
Okay.
growing up. And this victorgorgia is growing off of it. Actually, what you could probably do is try to pick it up by the victorgorgia, which is flexible, and the rest of it might just come with.
Huh.
Yeah, that's actually a really good idea, Megan.
And you don't mind if I like squeeze the victorgorgia?
Nope,
Yeah.
it'll be fine.
Okay.
I'll just grab the whole thing. You got the big box open.
I'm gonna zoom
All
out.
right.
Yep, come on.
Slowly.
Nice.
This is going in the starboard box?
Up to you. We have a big box open forward and starboard if you're wanting to just get it in the
Rather
forward.
than forward, yeah.
Yeah, so
Just 'cause
forward lambda would be
This
good.
could fall apart.
Goes down. All right.
And
Forward box open.
And
Is the camera retracted?
Yep.
Okay.
the lambda, we have some brittle stuff in the omega.
Okay.
Okay. There we go. Just wanted to make sure there were no floaties.
Is it gonna fit?
Oh, it's gonna fit. I believe. I have faith.
That's a lot of confidence there.
Is this the same one we were trying to collect yesterday? Or
No,
similar?
this is different. So the one we were trying to pick up yesterday was
The yellow one?
Samia,
Oh.
and this is a Madrepora.
And victorgorgia.
And victorgorgia with a bonus. Actually, the
Got a real
victorgorgia is
start.
what made this whole operation possible.
Well, it is a little tall. I thought it was gonna go sideways.
Those polyps might just push down the victorgorgia. I don't know.
Yeah, I think if you just close the box, it'll...
Looks
just
like the
fold
yellow
over.
part is all the way down in there.
Little bits. Little bits.
Yeah, I think it'll just push down.
Yeah.
Yeah.
Nicely
Nice.
done.
Perfect.
That was
Yay.
spectacular. The mastermind, Megan, coming out with a master plan, and
Yes.
Sarah and Jake just, I mean, that was
Executed
phenomenal
it.
teamwork, and obviously Aaron being our eyes, that was just spectacular. Thank you very much.
Awesome.
What's
Well
the
done.
sample number?
This is number 86.
86.
Yeah,
Yeah.
good work.
Next, a rat tail.
Yep.
We're just warming up.
That's
Yeah.
practice. Good practice.
Oh, what's this little pink? Oh, it's a shrimp, I think? Okay.
Ooh.
I thought it was something else for a moment there.
Good?
Oh, yeah, I'm good. It's
Oh.
just a shrimp.
Oh. Oh, little fish.
All right, we're approaching our 1,000-meter mark. We'll do our next Niskin sample.
Is that a balloon?
It's
Oh,
fast.
it was an Acanthophira
Oh,
type
okay.
of shrimp.
I was like, that looks like a balloon. Oh, this texture is weird.
We're starting to see numbers increase a little bit
Uh-huh.
as we keep waking ourselves, moving ourselves up this seamount.
Against the current.
Wait a second. I'm yanking on Atalanta a little bit. Oh, what is that guy? Probably a little shrimp. Or it's just a shadow.
There's a fish there.
Uh-oh. And go in there. Let's see if I can keep it a little steady.
It's
Oh,
going to be
no,
a bit
it's
difficult.
not a fish.
No.
I was
And
gonna
it's
say.
just a rock.
Uh,
Oh, we were both convinced. I was
I know.
seeing a shadow, but it's just two dark lines on the rock.
yeah.
Yeah, I thought it was a shadow too.
There's
maybe
a
it
little slime
sm-
star.
yeah, I think that is new for this dive, at least the portion I've seen.
Let's see.
Oh, that's a nice urchin.
Yeah, nice pencil urchin.
Mm.
See if I can keep her grounded. Go in there.
Is its stomach out?
I don't think so, but
It looks like it'd be so squishy.
Yeah. And slimy.
Oh, it's definitely squishy.
And it is slimy.
Yeah. Yeah, this is a pteraster.
I wonder if any people eat these sea stars
I don't think
besides
people
Mario.
I don't think people eat sea stars.
Huh.
They don't make for good eating. Not a lot of nutritional value. Kind of like sea cucumbers. There's just echinoderms don't have that many like parts that are really edible. Like urchins, what we eat the gonads, but we don't really eat the rest of the animal. And like you said, the sea cucumbers, you eat the gut. And there are some shallow water sea cucumbers that people eat whole.
Yeah.
But in general, only a few species are consumed. But I've never heard of any sea stars that are food.
You can push out.
And we generally don't see a lot of sea star predators.
Sorry, are you saying that there's not a lot of organisms that eat sea stars?
Right,
either?
not that I can think of.
So are they at the top of their little
Can we
food
take a look
chain?
at the yellow sponge?
Over on the right here.
This one?
In some places, yeah, the sea stars can, really run the community.
Oh, this is a cool color sponge.
Yeah, this is different. So remember when I was like super convinced that the sponge was yellow and then it was zoanthids? But now I feel like this sponge is actually yellow.
Might just have to try fly in here. Find a little ledge to pop on. Oh, all right.
But there are zoanthids on this sponge. But they're not what's making it yellow.
All right. Let me tilt down a little bit and we can go in on that.
Are those barnacles growing on it?
There's zoanthids.
Oh.
Yeah. They were open before, but they closed up because they felt the current thruster wash. I think this might be a demisponge of some kind. Yeah. That's cool.
Can come out. Thank you.
It's a
Here,
SpongeBob sponge.
I think we collected one of these yellow sponges on the 167 expedition. There was a lot of like this yellow sponge around.
Yeah, we have a little bit of one.
I have a comment here in our scientist ashore chat. Thanks, Sako, mentioning that even the Japanese don't eat the sea star.
Ooh,
Oh,
jelly.
there was another one of those cucumbers,
Translucent.
the
Oh, it was a sea
Pelagipteria.
cucumber.
Yeah.
This little pink one? Is that?
Oh, there was one of
No, it
those
passed by.
pelagic sea cucumbers that passed by.
Oh no.
It's okay. It
Missed.
was going really fast.
Have you guys tried sea cucumber?
I have not.
Huh. Interesting.
I've been kind of afraid to try it, actually.
Oh, yeah?
I really want to
I'm
try
worried
it.
it's going to be slimy and crunchy.
So, the ones that we eat the intestines, is kind of slimy. But then there's the ones that you eat the skin, and it's kind of processed in that way. We usually put some Kool-Aid on it. You know those
Kool-Aid?
small packets of Kool-Aid, the unsweetened ones? Yeah.
That's interesting.
give
I
it
never
a-
would have. I never would have guessed.
Gets it a little sour.
But I thought you were going to say like, oh yeah, we put some soy sauce on it or something.
Yeah, even soy
It's like, oh yeah,
sauce.
Kool-Aid packets.
Yeah, Kool-Aid is its own
Let's take a
seasoning.
Yeah. They tend to avoid putting lemon because it makes it harder. It makes it like a lot
Like firm.
tough. Yeah.
Oh. Oh, interesting.
There
Oh,
we go.
there's a little anthomastus. That's what that is. And the basket star.
Can go in.
So pretty.
Do you put Kool-Aid packets on anything else?
Like, you know.
Yeah, there's the last expedition. There was some spice mixtures made by one of our previous scientists from Pickerick, Maikani, with Kool-Aid and other spices, and we were dipping it in fruit.
Yep.
Oh, kind of like a leaking.
A salt. Yeah.
Kind of
Mix it with
situation.
salt.
Yeah, kind of.
Cute.
That's beautiful.
Yeah, it's really pretty.
They also there's the kimchi, aji kimchi. They would
It's
just
very
mix it
relaxing
with
to watch.
That sounds
Kool-Aid.
good.
Yeah.
But with Kool-Aid?
Yeah.
Can come out.
Is this the base?
Is it what?
Is that the base of the, or is it?
Oh, so it's a dead
dead-
sponge,
dead,
and that's a
yeah.
basket star.
Yeah.
That all that like hair, it's one basket star.
I mean the little, that part.
Oh yeah, yeah. That's the base of the.
Of the anthomastus.
Yeah.
Yeah.
It's beautiful.
Yeah, that was a really cool shot. Thank you.
Yeah.
Kind of reminds me of a cacti with the little flower, the single
Oh,
flower.
kinda, yeah.
There's this. Sorry, I'm still talking about Kool-Aid.
Yeah,
Go on,
I
go
want
on.
to know more of that. Go on.
There's this
Yeah.
trend of these kids. They add it to like the sour gummies.
Ooh,
Okay, yeah.
That
and
sounds
some
good.
actually add it to like hot Cheetos
Hot
as
Cheetos.
well. Yeah. Kind of like spicy.
Spicy
can't-
and
yeah.
fruity.
This is making me really hungry.
I'm just like, I have nothing to compare like a sea cucumber to, so I'm trying to think like oyster, you know, where
No.
with the goo and then, I don't know.
It's a little different. It's kind of
Jello plus fish.
We're gonna have some waiting for us once we get.
We'll try some Palawan delicacies.
Yeah.
But other islands in the Micronesia, they like to eat it with. They would eat it with rice, that's bizarre to me. They would add it with their meat to give it that tangy taste.
See, that's what I thought you
With,
were gonna say how you ate it, was with rice. I
With
don't know
rice,
why, but that was my default.
with rice, is that what you said?
Like they eat it, they mix it with the meat, and then they eat it with rice. And
Oh, okay, yeah.
Yeah.
It's
Yeah, that's definitely how I imagined it as well.
So we are.
to me that's weird.
It's
So-
really interesting.
Yeah, we're crossing here 1,000 meters. Should we go ahead and take
Oh,
the
yeah.
eDNA
let's take
sample?
that
Let's do it.
sample.
Sample it up. Number 2.
We're right on the
Here,
mark.
I can hold. Find a little spot to perch on real quick.
And as we set up here, I think one of our science leads ashore is also on the line. He can allow.
Can also do it in the water column.
Yeah, we're gonna need you, Igalo, to
Yeah.
find some
Hey,
sea
can you hear
cucumber
me?
for us.
Hi, Kilo.
Yeah,
Hi.
loud and clear.
Just
Hi, how are you guys doing?
Doing
see
great.
if I can stay right there.
Getting ready for Niskin.
Right on.
Which one is this?
Number
These have
2.
been some,
See if I'm retracted all the way. There we go.
Yeah.
So for the viewers, we have these containers on the
Alright,
side.
fired.
Once they pull that, it closes up, it traps the water in the Niskin bottles. And then when Hercules makes his way up, we have the fun job of getting those waters.
Thanks, Jake.
Can
Thanks,
we look
team.
at the pink sea star below you?
Absolutely.
Pink sea star.
While we get set up here, maybe Icalo can tell us a little bit about the eDNA program and all our efforts by Pickrick.
yeah, thanks, Daniel. So eDNA started in 2001 at Pickrick in collaboration with Stanford Center for Ocean Solutions. And it's been a great opportunity to be able to get a long-term time series of water samples around Palau.
See if I can settle
So
out.
we've been going on for about 3 years now. And what's great about it is we've been able to go around Palau in all of the directions. So the Nautilus actually is giving us, is able to expand our
going?
scope of collection. So in the past, we've only been able to go so many miles off the main island. We've never been able to come down this far to the southwest. And so these surface samples, as well as ROVs, because all of our past collections have all been on the surface, and environmental DNA degrades with UV light and the time and the amount of time that is spent in the water.
Doesn't look like the
So
perfect
collecting them
pastry
deeper.
one. Ooh.
Collecting these samples deeper gives us a better understanding and possibility of better diversity of what's in the Palau's EEZ. We're super grateful for this opportunity.
Yeah, thank you, Icalo, and we're very grateful for all your help in helping plan and execute this mission. I know it's
Well, thank you.
a little bit different doing it from shore, but really appreciate you joining us both day and night and all the support from shore.
My pleasure. It's been a blast watching the live. All of the dives have been amazing, but I have to say that the Southwest have shown some amazing cool critters under there, and even the topography of the seamounts. I'm pretty stoked. I'm geeking out over here.
Awesome.
What has been your favorite thing that you've seen so far?
Um, the Dumbo octopus
Yeah.
had the top,
Yes.
and just about an hour ago, probably that sea cucumber.
It's a really small jelly.
So I'm like over here Googling everything about it.
How did she connect the...
The wonderful telepresence technology. So yeah, for viewers on shore, so we have, obviously we stream the video and the audio here from the control van on EV Nautilus. We also have a chat basically that connects, and for this expedition we got close to 90 or so scientists that are watching, but they're also talking to us via chat base, and then we also have the way to patch in via audio. And Ikelau has been joining us in both day and night odd hours. I really appreciate all the support.
Yeah, I got a question from the chat, kind of regarding the eDNA. So they're asking kind of what kind of data has the eDNA shown or provided so far since it started. I didn't know it started that long ago. Maybe.
Ikelau, if you're still with us, you want to take that one?
Yeah, sure. So the thing about eDNA, it takes a little while for us to process samples, but from the data that we've collected so far, it's been able to confirm the like yellowfins and mahis that we're seeing in the water. But one thing that was really cool that stood out was it picked up some of the mammals, so we were able to see some of the whales that have. There's been reports of them in the water, but no real actual sightings. So we captured that. And then earlier this year, we went and we did a cetacean survey, so whales and dolphin surveys, and we were able to visually reconfirm our eDNA indications. So that was really cool.
Don't think I'm gonna get them.
That is really cool.
Yeah, thank you.
Try and zoom in there. Oh, he's on the bottom too.
He's like struggling to move.
So tiny, so fast.
Yeah.
Is this one of the little, I mean, no, it's not red, so it's not a little red jelly.
It's kind of red.
It's kind of.
Is this the one that the paper was talking about?
No, not that specific.
The tiny little jellies.
Little red jellies.
The little red jellies were all the little red jellies on off the California coast.
Oh,
that was really what the paper was focusing on, but a lot of those jellies are found a lot of places in the Pacific, and they're very cryptic. They all look very, very similar, and it's hard to get good imagery of them because, as you can see, they're quite quick.
Yeah.
And they're very small, and it's hard to sample them as well because they're very fragile and small, and they're hard to preserve. So getting good imagery is the best way to identify them, but that's also an extremely challenging task.
The HD is a bit shaky. Especially when I'm lateraling to port.
let's go and look at that sponge straight ahead.
Yeah. Stretch us out a little bit.
Oh, I don't know if you'll make it. Oh yeah, it's that like super branchy sponge. I thought it was something else from far away.
Oh.
But these sponges have a lot of associates with them. They make really great substrate for a lot of animals to live.
Oh, what is that moving?
3 different kinds of crinoids on this sponge. Some ophiuroids, pearl stars, all over here.
Can zoom in.
What?
Go down over. These guys.
So we have this little Crotallometra feather star. There's a bunch of amphipods as well. Some shrimps,
Ooh, I see a
anemone.
shrimp.
Yeah, there's a lot of associates up in here.
And the off-colored ones, are those the dead ones?
Yeah, where it's yellow, it's dead branches.
Huh.
All right, can come out. Yeah, it's real shaggy.
Yeah, thank you, that was great.
Yeah.
Yeah, for those folks that just might have joined us, we just crossed 1,000 meters. We are about halfway through our last dive of the Lebus Voyage 2 expedition to the Palau National Marine Sanctuary. We started the dive today at 4 a.m. Palau local time, about 1.5 kilometers north of where we currently are, about 500 meters deeper than we currently are. And we are moving up a ridge arm, if you will, towards the summit of the seamount. That summits at about 760 meters or so. And the plan of today's dive is to climb up this ridge to the top and then spend the last few hours moving across the top of the seamount where we have a pretty well-defined ridge. So far, the animal counts have been quite a bit lower than in the last 2 dives. That's probably related to this being less of a favorable environment. For those folks that were with us on yesterday's dive, that was really an extraordinary community of deep sea organisms. So So a lot of exploring still to go. And yeah, Oreo, large Chrysogorgia. Yep.
Looks pretty cool.
Is there a basket star on that Chrysogorgia?
Yeah, I think so.
There was a little tiny fish too.
Not the Oreo?
No,
Another
little.
one.
Zoom in.
Is this?
Oh, there's 2 basket stars. And there's a shrimp on the basket star.
That's on the
It's on a Chrysogorgia.
Chrysogorgia.
I really do enjoy seeing the basket stars.
Oh yeah, they're so fun. They're just like the way they curl and uncurl. Like you could watch this all day. It'd be a great screensaver.
Yeah.
Right.
I think it kind of freaks me out.
It's just a little bit uncomfortable.
Yeah. I feel, I don't know.
And the shrimp.
It just always impresses me that like they're not all like tangled in knots all
Yeah.
the time.
They know exactly where they need to be.
Just feels cluttered, I don't know.
All right, can push out. Yeah, that's a lot of fun.
That's pretty big.
Beautiful, yeah. Let's do a staff soon. I think that's a Leopathes, new for this dive.
Quick zoom. Settle down.
Yeah,
there
there's
we
a
go.
bunch of these sternostylus squat lobsters.
Find a little perch here. Can go in on that.
Here, just for you, Elsie, since I know you enjoy names now, so these are Leopathes. So pathes again for the
Black coral.
for the pathogen inhibiting substances that are thought to found in a lot of black corals, and Leo being smooth in Latin. And though this is one of the most primitive of the black corals, one of the earliest originating black corals, if you were to remove the tissues and remove all that bright colored and see the skeleton, it would be black. And then if you look on this under a microscope, it would have tiny spines, and that's what characterizes black corals. They all have these tiny spines, except this group of black corals. They have, in some species, those spines are completely gone, and others they are reduced to small bumps.
Hence smooth.
And this is probably, it might be, but if not, related to it, Leopathes annosa, which is Latin for long-lived. And this is because individuals of this species have been dated to be over 4,500 years old.
Oh, wow.
So making these individuals the same age of, or having in some cases been along longer than when the pyramids were built, or when the first ancestors of Polynesians started going across the Pacific. So very, very different time scales for some of these species.
It's incredible. And the dating is, you would have to collect a sample in order to do the dating. You can't do it visually.
Yeah, that's correct. And you would have to take an entire colony and use the base. And basically these things, just like terrestrial trees, you would cut the base in cross-section and then you would see growth rings. Now those growth rings are not necessarily annual. They're not, they're just times of faster and slower growth. And what is done with these very slow-growing animals is that you have a technique called bomb carbon. So there's different isotopes of carbon, including some that are naturally occurring and some that are non-naturally occurring, the not naturally being a product of atomic tests done in the 60s and 70s. And so there was time in the 60s and 70s where there was an unusual amount of this bomb carbon in the world, and that was taken up by a lot of animals all over the ocean. And so if you basically take that cross-section and do very, very fine measurements from the outside going in, at some point you will detect the signatures from those bomb testings, and then you can calculate from the very outer inside how much time, how much growth has happened. So now there'll be a certain distance, and then you can extrapolate that to the center and then get a sense of the growth. So obviously not a perfect measurement. There's some error associated with it, but it gives you a good rough estimate of the ages of these animals.
There's a star right next to, is that normal to have more than 5 tentacles or 5 arms?
Yeah, there's some that have 7, 9, and others, yeah.
I think we saw that one earlier.
Yeah.
It's just shining really bright.
Oh, fish.
Oh, fish. Or eel.
It's an eel.
Eel.
But what kind?
Yes.
It's a cutthroat eel.
Oh, this one's kind of long.
You can zoom in there. Oh, maybe.
It's eating something.
It's quite big, yeah.
So this is Synaphobranchus affinis.
That's a nice shot.
And a furriid.
Oh.
A furriid. Is that a furriid?
Yeah, it is a furriid, and it's got some, I think, zoanthids on it.
Oh.
Into the ground. come out.
There's a paragorgia.
And a chrysogorgia?
They seem like very sporadically spread out.
Yeah.
It's kind of here and there.
Yeah, now we're starting to see a lot more of these glass sponges with those crinoids on top that we... 2-3 dives ago, that was the dominant organism as you'll start going up into the top of a seamount. I wonder whether we will see the same.
Has the current changed?
Seems like it isn't. It's calmed down a little bit. I haven't tried going straight lateral to port yet, since we've been kind of having with the angle, kind of more being more starboard. But it does seem, the thrusters definitely seem to be putting some work in.
The surface currents changed a little bit for the ship, so. I don't know if you noticed the bow thruster kick in.
Yeah, we've been pretty fortunate over these last 3 weeks that we basically had spectacular, absolutely just magnificent weather for the first 2 weeks. I mean, I haven't seen it that calm. We really only have one day where it was just really the misalignment of the winds and currents that prevented us from deploying.
Oh, there's a fish.
Is that?
Little rat tail.
Huh.
Oh, good eye.
Huh.
Can zoom in there. Oh, maybe. Too fast, too fast.
Oh, really fast.
Oh, no.
Very fast.
See.
Oh, it actually might be a Castillo, now that I see it.
Yeah.
And more of that rubble, coral rubble.
Yeah, we've seen this on a lot of the seamounts, but not a lot of actual big amounts of coral.
Colonies.
It's really interesting.
At one point,
Cool, thank you.
they must have been
Can come
abundant.
out.
Oh, crab. Or Is that
Oh yeah, it is a crab. Look at that.
Oh yeah.
Check that one out.
Oh, oh,
where are you going?
here we go, here we go.
So fast.
Ran right underneath me.
I knew what I was doing.
Yeah. Then it's like, I just freeze. You can't see me if I just hold still. See if I can get in there like that. Take two. Oh, he's already go.
Is this another golden crab?
I don't
looks
think
different.
so.
Looks different.
Yeah, I
I
don't
think
think
it might
Try
be a
and
homolid.
zoom in there.
Molid.
Look at that.
What is that?
Is that a siphonophore?
Yeah, there's a siphonophore right there. Yeah. Oh, it's one of the carrier
Siphonophore.
crabs, but this one's not carrying anything.
Oh.
Oh,
Oh.
he's looking for
Something to
something.
carry.
Oh no.
Go.
Oh. Searching for a treasure.
Yep.
Oh, and
Oh.
there's a
There's a jelly.
Oh.
jelly. A lot of these just coming up in here.
Yeah, there's a Paralia, that's the jelly. And then
Probably heard we're about
go
to.
take a look at that siphonophore.
Mm,
I think it's going to line up really nicely for you. You can see the swimming bells at the top of the
Can
screen.
we get lasers off, please?
Yep. Oh, there. Thanks, Jake. Can go in on there.
swim towards the blue. Wow. That is unreal. So this is a jellyfish?
It's a siphonophore, so it's not a jellyfish. It's actually a colony of animals that all have their own specialized functions. So they work together to form this colony. You have ones that specialize in swimming, ones that specialize in eating, ones that specialize in reproduction. And so these are, if you're familiar with Portuguese man of war or blue bottles, this is in the same group.
That's pretty interesting. Oh yeah. Well, starting to get all exciting by the time we have to change shift.
Can I just see the
Well,
Niskin?
we can still watch from the lounge.
Yeah.
Which is what we usually do until
Yep. We
Until we
don't
go
watch regular TV. We just watch the dive.
Right, so to our viewers, we are going to be doing a watch change pretty soon. So if you look at satellite feed 3, you'll see the new watch team coming in.
Check
You'll
out
see
that coral, Sarah.
oh yeah.
the current
pretty.
team kind of brief everybody that's coming in to.
This is like a fan coral.
Yeah, it's a primnoid coral, probably Paracalyptrophora.
I really enjoy looking at these ones.
I'm switching. Or do you want me to zoom in before I switch?
So yes, thank you for
see if I can
joining us. Make
set
sure you
it down
continue
here.
watching, and we'll be back at 12 midnight
Yep, for the last watch.
for the last watch. Sounds so sad to say.
I know. It seems too soon.
Thanks, everyone.
Yes, thank you.
Hold over a little bit. No zoom. You want me to wait for zoom or?
Oh, we can go in.
Can we zoom in? I don't think she's ready.
Oh yeah, whenever they're ready. I just thought this might be a fun thing to look at while we
Yeah.
switch around.
Hey, video. Wait, what's her name? Hey, can we zoom in? Thank you.
Sorry, I just put my headset on.
No, you're good. I saw you. Awesome.
Yeah. Beautiful. I love it.
So,
Let's go. Where are we going? Where are we? Right there. South down. Map down right there.
all right, welcome back.
Give me 1 second here. Totally stoked.
All right, welcome back for the 8 shift. This is our last shift, guys.
Is it? No, it's not.
Is it?
No.
I thought it was.
No.
Yeah, not for you up to map, but the rest of us.
No,
Oh,
are we
yeah,
picking
yeah.
up?
At
Oh.
2.
Oh, okay. For some reason, I thought we were picking up. Oh, all right. Yeah, it is. Cool.
Oh, we recover at 2?
For some reason, I thought we were recovering on our watch.
That was the original plan.
Oh, it was the original?
Yeah.
Okay. That's. So I'm not just making things up. That's good this time.
You just are behind the times.
That's fine.
We have a new plan?
Fish, please.
Right there.
Yeah, I heard that we're bringing Hurt back up at 2.
Yeah, recovery is 2 a.m.
Oh. So coffee in the data lab this morning. That's my future.
Did everybody nap pretty good today?
Oh, yeah.
Oh, yeah.
You ready to start moving, Dan?
We're going to zoom on the fish.
Are we going to play around here for a little bit?
I have no idea.
Do we want to move? Anybody want to move?
Generally in favor of moving.
All right, let's move.
Quick look at the fish and then let's move.
I'm going to start moving, Dan.
Roger. I'll start following you.
One way or the other. Bridge, nav. Let's do 30 meters, 175.
Okay, you can go.
Very good. Thank you, Ariel.
And the uphill seems to be this way. Immediate locale, at least.
What? My phone is dead.
Mine's not. Yeah. Thank you.
Here we go.
All right. All right.
Yep.
You go, Amber.
Maybe have to do it twice because I
You usually do.
I had to forget.
It's still blinky blinky.
Yeah.
It's such a pain. There is a.
Got it. Maybe.
You got it? All right.
Maybe.
Okay. Ooh.
It's sinking about it.
Here's another Oreo.
Those aren't Oreos, those are Starburst.
No eating in the van.
Screen.
Do as I say, not as I do.
Can we zoom coral while we're right here?
Yeah. No eating of crumbly things in the van.
Yeah. Chewy, sticky things, no problem.
There's still M&Ms rolling around in here if you look closely.
Yeah.
As long as you,
They say
as long as you
those
mute your
M&Ms
microphone.
are still rolling on the floor to this day.
You can zoom in there if you want. Yeah.
It took
Interesting.
This looks like that planar Chrysogorgia we sampled a couple dives ago, or something similar.
And then down next to it is that hydrocoral?
Stylastrid.
Stylastrid.
That's a stylastrid to the left and a cup coral to the right. Right, we're good here, thanks.
Okay. Blinky blinky.
Quinlan, can I see the sample sheets real quick? Thank you.
All right. Welcome back to knowledgelive.org. This is the 48 shift. We'll do introductions, and we'll start with Taya. And since this is our last dive, I'm curious to know how each and every one of us in here how did we get to this position? How did we get to be a part of this expedition? So maybe a brief explanation of that.
So my mother is a really passionate scientist, so I was lucky enough to kind of inherently get the opportunity to come on this trip. But I didn't do anything personally. I was just caught in the very lucky crossfire of it. But yeah,